Hello, I'm Ed Moriarty, Director of Worldwide Marketing at TA Instruments. I would like to welcome you and thank you for joining us in today's webinar. If you get disconnected at any time, please use the instructions you received to log back in. You can access various content by clicking the Documents widget at the bottom right-hand corner of the screen. This includes speaker bio and additional file downloads. If you need help at any time, click the question mark widget. Please ask any questions you may have at any time during this presentation by submitting them through the Q&A window. We will answer as many of these as possible at the end of the webinar. Our guest speaker today is Dial Holton. Dial earned his PhD in immunology and microbiology at the Medical University of South Carolina. He has 20 years of experience in the field of microcalorimetry and has been the worldwide microcalorimetry product manager at TA Instruments for over 8 years. Over his career, he has published over 20 research and assay development papers in the areas of cancer research and biopharmaceuticals. His experience in both academic research and industrial development laboratory environments will bring a perspective that will include the newest ITC and IMC techniques, as well as instrument innovations that ensure the highest data quality when developing and characterizing new drugs and biopharmaceuticals. Today, Dial will be describing a new and recently published ITC technique called enthalpy screening. This new technique is perfect for rapidly and accurately characterizing the binding enthalpy of new drugs or biotherapeutics. This rapid screening technique, coupled with the fully automated Affinity ITC instrument, shortens the time required to produce the powerful thermodynamic signature for any biotherapeutic being considered for further development. Dial will also be discussing the power of isothermal calorimetry with some preliminary feasibility data generated on the flexible and powerful TAM4 instrument. This technique promises to dramatically shorten the time it takes to determine the accurate shelf life, kinetics and aggregation kinetics for protein formulations, and even new high concentration biotherapeutic formulations. The title of his talk is Characterization of Drug Binding Activity and Biopharmaceutical Stability. Dial?
Thank you, Ed, for that introduction. I want to welcome all of you to this webinar on the characterization of biopharmaceuticals. I hope today I will be able to give you some preliminary data and some insight into the use of microcalorimetric techniques for characterizing biopharmaceuticals and the explosion of biopharmaceuticals as we know them today. One of the things that I typically start out with is a definition of a biopharmaceutical, and this is something you can look up on the internet. It'll give you a lot of different definitions, but basically it's any pharmaceutical drug product manufactured in, extracted from, or semi-synthesized from biological sources. They can be called a biological medical product, a biological, or just biologic. So if I use these interchangeably, you'll know what I mean. The idea of a medicinal product or a therapeutic that is a biological is specifically engineered from macromolecules like proteins or nucleic acid-based products. It's distinguished from blood or blood components or vaccines, which are usually extracted directly from blood or from a biological source. Here's an example of the different major kinds of biologicals that we might be looking at. We're going to focus on a very small number of these, but looking at blood factors, hormones, interferons, all of those can be classified as biopharmaceuticals. The one we're going to focus on today is in red, the monoclonal antibodies. One reason is that they are active. They bind to their target, and we can measure that. It's something that we have instrumentation that's been developed recently that will allow us to do that very effectively and very rapidly. Here's a list of several of the newer biopharmaceuticals that have been introduced just in the last few years. You'll notice they range from treating rheumatoid arthritis, things like anemia, things like psoriasis, breast cancer, all of the different kinds of diseases where you can use a biological product and have an impact on the disease state. These are the classical kinds of biopharmaceuticals we have today. If you look at the top 10 drugs back in 2010, you would have noticed that there would have been several in the top 10, but leading the way are the cholesterol drugs. You look at the drugs today, and even in 2014, this list is out of date by a couple of years, but the idea here is that 8 out of the top 10 are biopharmaceuticals, and that's only going to increase over the next few years. If you look at the use of biopharmaceuticals as well, as the number increases, the use is going to increase. And this is a set of data, a graph that shows the use of either small molecule drugs or biologics. And you'll notice that the biologics are on an upward slope, and we are using more and more of them, approaching 25%. And you'll notice also that the small molecule drugs are decreasing in the amount that are being used. So as the biologicals increase, the small molecules are going to decrease a little bit. And that's something I think we at TA Instruments looked at and decided that some of our instrumentation can have an impact in the development of those biologicals. So what are we going to talk about today? Well, I'm going to talk about 2 different sets of data. One is an enthalpy screening with ITC, isothermal titration calorimetry. It's a direct measurement of the binding enthalpy using isothermal titration calorimetry, and you combine it with some additional data to get a full thermodynamic signature. The second set of data I'm going to talk about comes from looking at isothermal microcalorimetry. This is something done very effective on a TAM instrument, a thermal activity monitor. It is an isothermal measurement that is unmatched in its sensitivity and flexibility, and it's a direct measure of the heat flow from either a dilute solution or, more commonly these days, a very high concentration biological. And you could rapidly estimate the formulation stability and the shelf life, and to a certain extent, the aggregation potential that's going on in a biological as it's sitting in a static formulation. The first instrument that we are going to talk about is the Affinity ITC auto instrument and the technique of enthalpy screening. This instrument was released in 2015 by TA Instruments, and it's a fully automated ITC. It's something that is fully programmable, and the experimental design lends itself to easily setting up and executing an enthalpy screen and with using minimal reagents. Up to 30 ligand screens at once can be loaded into the injection syringe, and then by just changing what's in the cell, you're able to quickly go through and screen the enthalpy of the binding reactions. It's fully automated in cleaning and rinsing, so that if you have a problematic sample, one of them that perhaps you want to make sure that you've cleaned the cell extremely well, it's easily automated and can be executed anywhere in the experimental protocol. The other thing that TA likes to do is provide software that allows the user to be able to analyze the data rapidly and accurately. And with nanoanalyze and batch processing, once you've generated all these files on an automated ITC, you can then easily go through a batch process and analyze and determine the enthalpy with an enthalpy screening model. So what is the method that we use for enthalpy screening? Well, first, you have excess compound or ligand that is placed in the reaction cell. This is just the opposite of a typical ITC experiment where the ligand's in the injection syringe and your target molecule is typically in the cell. The concentration of the compound is usually about 100 times the IC50. Now, it doesn't have to be exactly that number, but that's an estimate in thinking about experimental design for an enthalpy screen. And then you inject very small amounts of the protein or the target into the ligand. This is going to give you the maximum enthalpy, and you can get that maximum enthalpy in more than one injection. And so you make at least 3. And what you see on the right-hand side of the screen here in the graph, you'll see that at least 4 injections were made. The first one is a very small one, and then 3 identical injections of 2 microliters were made, and you can see that they are identical. By averaging the enthalpy values under those injection curves, is that you're able to get a very accurate estimation of the enthalpy of that binding. So why would we want to get that? Well, more and more, the thermodynamic signature is becoming something that is a powerful tool in characterizing the binding of a drug or a biopharmaceutical. The idea, as you see on this graph, is the enthalpy, or delta H, represented by the green bars, is that you're measuring the hydrogen bonds or van der Waals forces, or even some polar desolvation if you actually get an enthalpy that works against the binding. But understanding whether the binding or the delta G, the actual energy of the binding, is actually driven by enthalpy, or is it being driven by the entropy value, which is the minus T delta S? And by determining that enthalpy value, you can then solve the equations on the right-hand side of the slide, understanding what delta G is, and then calculating what delta S, or your entropy value, is. So why is that important? Well, back in 2008, several publications came out showing that different drugs that were developed at the time, as they got better and better over the years, is that they became selected for what was the driving forces of enthalpy. So the better the enthalpic value was for each individual drug, the more effective the drug was and the less side effects that were encountered by the patients taking them. This is just an example published in 2008 of the statin drugs. And the statin drugs, as they became more and more effective, is that they discovered that we were selecting those drugs because of the enthalpy value. The enthalpy value, the driving forces, the specific binding of that drug to its target was something that was giving us a more effective drug. The idea that the enthalpy screen value is the same as what we might get with a standard titration is shown in these 2 graphs. This is extracted from the most recent analytical biochemistry publication from Johns Hopkins University by Ernesto Freire and Arnie Schoen, showing that if you look at the 2 examples that are shown, the ones that are using KD on the left-hand side are the thermodynamic signatures that were determined by using a standard titration. And the 2 graphs on the right-hand side of each one, as you'll see, was determined by an enthalpy screen. And despite any minor numerical differences that we might see on these 2 graphs, the binding characteristics of both of these drugs are well defined by both of these methods. So the enthalpy screening, combining it with IC50 or Ki, as well as understanding how to calculate delta G and minus T delta S, gives us a good understanding of what's happening with the thermodynamic signature. In that same paper, they looked at several HIV kinase inhibitors and trying to understand if by enthalpy screening they could get the same values that they would get if they had done a full titration. The full titration data had been published previously back in 2008, and the idea was to create the same kind of thermodynamic signature. And what you see here are those drugs as they are plotted out, and you'll see the differences in the enthalpy values, the differences in the entropy values, and even one on the right-hand side that shows that the enthalpy value actually works against the binding. And if you compare these with those values that were determined by a standard titration, you'll see that the correlation between the 2 is at least 0.96. This is something that trying to get a correlation between the 2 and making sure that the enthalpy values aren't biased in some way because we are not doing a full titration. So all of the data seems to point toward the idea of an enthalpy screening being rapid, it's accurate, and it's correlatable with the values that have been published that have been determined by full titrations. The enthalpy screening with ITC is that powerful technique for rapidly and accurately characterizing the enthalpy. The typical enthalpy screen requires less material than a conventional ITC titration, much less. The Affinity ITC auto instruments, as the automated ITC instruments have been introduced, it's innovative, it's reliable, it's easy to use and set up an enthalpy screen so that you can perform them rapidly and easily. The binding enthalpy measured by the ITC enthalpy screen allows for a good approximation of that thermodynamic signature of any molecular interaction, especially with biologicals. And enthalpy screening provides that critical information for more informed candidate selection decisions, especially in the early stages of drug development, where you want to be able to look at and determine which drugs or which biotherapeutics to carry forward in the process. So I hope that's given you a sense of the excitement that we have around enthalpy screening using ITC at a very early stage of selecting of candidates as for biopharmaceuticals or for drug candidates, and the idea that the instrumentation is there to help you do it. The second set of data I'm going to talk about, once you've made that candidate selection and produced some of the biopharmaceutical, is what is the formulation stability and how can I analyze that quickly and accurately without going through a full, sometimes multi-week, multi-month shelf life study. And the idea is that virtually all chemical processes, whether they're in a biological or in a drug, result in either heat production or heat absorption. And the idea that denaturation actually creates heat and even aggregation will create heat in such a manner that you'll be able to measure it, whether it's being absorbed or whether you're getting an exothermic event. Calorimetry qualifies the amount and the rate of heat released in terms of heat flow, heat, and heat capacity. And this is important to know when you start characterizing things that have to do with compatibility, that have to do with stability, that have to do with aggregation. Calorimetry is also a non-specific technique, making it ideal for studying the kinds of physical and chemical processes in life sciences, material sciences, and within the pharmaceutical field, and especially the biopharmaceutical field. So in background for this particular technique, biopharmaceuticals are not typically orally bioavailable. You have to deliver them a different way. Traditionally, deliverable configuration is by IV infusion. This is something that sometimes patients really don't like, having to be hooked up to an IV machine and take the pharmaceutical that they are taking. However, more recent developments is that patient self-administration can be done, but it requires very high concentrations because of the body mass in a typical human, 100 to 200 milligrams per mil. And there are some issues with that we'll talk about in just a second. So these high concentration monoclonal antibodies, for example, all exhibit different aspects of aggregation. They denature at specific rates, and some of the high viscosity creates a problem for delivery when it comes to biopharmaceuticals. So the primary goal of it in characterizing a biopharmaceutical in these areas where you're having high concentrations is to rapidly determine the best buffer and the excipient concentration that maximize the stability and minimize the protein aggregation for at least 1 year at these required high concentrations. You can do that with a standard shelf life study, but then again, you're talking about months, weeks, or even months before you even get any data that you can count on for shelf life determination. One of the things about using a TAM, an isothermal calorimeter, is that when you start looking at heat flow, this is a classic kind of example where you'd say, well, what do you actually look for? Well, you look for more heat flow. And the one that produces more heat flow, the red curve in this particular graph, is the one that is the least stable. And the most stable one is producing less heat. So as we go through the examples I'm going to show you, hopefully you'll get an idea that as you start to determine how much heat flow there is, whether it's in a dilute solution, whether it's in a concentrated solution, you can get an idea of the kinetics and the heat flow that's happening within those molecules. The TAM itself is a flexible, multi-channel microcalorimetric system. You can run up to 48 individual and independent calorimeters, so you can get some higher throughput than typically you can get on any other calorimetric system. You can get down to the nanowatt range. So the idea that if you have a small amount of heat that's being generated in your sample, you're going to be able to pick it up in a TAM system. You can measure less than 1 milliliter or all the way up to 125 milliliters in sample sizes. You can have all kinds of sample handling systems, possibilities for adding and mixing, of looking at pressure changes, looking at dissolving solid samples, adding external probes such as light sources or pH meters. So the flexibility of the instrument goes on. And the new isothermal operating temperature range for the new TAM4, which was released about a year ago, was that you can get down to 4 degrees. So even those temperatures where you might not expect the heat flow to be very high, you can start getting down into the storage temperatures for biopharmaceuticals and drugs. So you have one system, and you have multiple possibilities for measuring the heat flow when it comes to biopharmaceutical formulations. So if you start thinking about protein thermodynamics, a biopharmaceutical, and you put it in a formulation and you want to start studying it, what happens? Well, the typical thing is when you increase temperature on a protein, you're going to start getting unfolding, and you're going to get an exotherm. As it continues to go, as you raise the temperature higher and higher, many times you're going to see that the protein is then going to aggregate and produce an exothermic event. And I'll show you a couple examples of that as we go forward. Here's an example that was published way back in 1994. The idea that you're looking at oxidation reactions and the idea that you can look at it isothermally at different temperatures. And what we did here was compare it to already published data that was generated with HPLC analysis. And this is something that more traditional shelf life studies where HPLC is used to measure the stability of the protein. And you'll see the correlation on the right-hand side. You'll see that it's very good. And as you see on the left-hand side, you can actually get down below that traditional 30 degrees C, which was used in the right-hand graph, get down to 23 degrees C, and even lower now with the new TAM4. And so as you go up in temperature on the left-hand side, you'll see more and more heat generated, therefore indicating that more and more denaturization or oxidation is taking place in the sample as you analyze it at that higher temperature. Another one is the stability of monoclonal antibodies, and this is a set of data that was generated on some monoclonal antibodies at different pHs. The idea was to look at the stability of them from pH 7.4 up to pH, or down to pH 3.4. So the idea is one pH more stable than the other? And as you see on the right-hand side, the red graph at pH 3.4, you get a higher heat flow or power in microwatts than you do at a pH of 6.1 or 7.4. So 6.1 is even lower than 7.4, and so you get a much more stable protein at 6.1 than you would at a pH of 3.4. And I think that follows traditional thinking around proteins and pH, is that depending upon the protein structure, it will have different stabilities at different pHs. So the isothermal calorimetry at 4 different pHs here, and this was published in 2010, is the idea that you can use calorimetry to predict that stability very easily. And in this particular case, you'll notice it's between 5 and 15 hours. So we're not talking a week, we're not talking a month, we're talking just a few hours to be able to get an idea if something's happening in the sample. This is a compatibility experiment with a TAM, just as an example to show what happens when you add an aliquot of water to 2 different samples that are combined. And this is looking at excipients. This could be looking at formulations of different monoclonal antibodies or different biopharmaceuticals. So the idea is that depending on the kind of molecules and formulations you have, is you're going to get different heat flows. And you'll notice that that top graph is the one where the water was added and the reaction created at least double, if not triple, the heat that we saw with just the 2 different dry components that were put together. This lets us analyze and say that the formulations that we originally had that were dry without the addition of water were more stable than the ones that when we added the water to it. So what do we do with characterization of proteins? Well, typically, if you look at a differential scanning calorimetric analysis of something as simple as lysozyme, and this is lysozyme at 150 milligrams per mil, is the idea that you get a transition and a TM at about 73.3, and that's classic for this formulation of lysozyme at 8.2. But you also get, as you go higher and higher in temperature, as you get up to about 80 to 85 degrees centigrade, you'll notice that there's an exothermic event due to the aggregation of the protein, and all of a sudden it drops off of the curve dramatically as it aggregates in the cell. So what do we do? Well, the idea here was to analyze the sample just before the transition peak, just before the 73.3 TM peak started. So as it initiated, we said, let's do some analysis at those early peaks and see if we can start to see kinetics of the denaturization and the kinetics of the aggregation as it goes further. So looking at that, we looked at 2 different temperatures. One was 61 degrees centigrade, and the other one was 59 degrees centigrade. And you'll see that there is a difference between the 2. The one at 61 is the climbing of the peak up to the maximum and then coming back down is a lot more rapid than what you see in the red curve. They do come to the same point further out at about 10 days. So as you carry it further and further out, certainly you're going to get closer and closer together. But that initial curve is something that is really important and informative as to understanding what's happening with not only how it denatures, but what's happening with the aggregation, if any is happening. So the preliminary data from that experiment alone seemed to say to us, okay, what's how can we model the denaturization process that's going on and the aggregation process? So we used a nonlinear least square fit that is for denaturization and aggregation. It's a model that's used. And we fit those curves, both of them, and we're able to show that they almost identically fit the data and gave a very good correlation. And looking at the DSC scans of both of pH 7.7 and pH 8.2, there is a little bit of a difference between the 2. So the stability of the proteins, especially at those analytical temperatures, may just be slightly different. And there are some slight differences. If you look at the graph on the left, the bar graph, you'll see that there are some slight differences between the 2, both in the rate for the denaturization that's taking place and the rate for the aggregation that's taking place. So early data said to us that looking at isothermal analysis of especially high concentration proteins could give us a very good idea of what's going on with how it denatures and how it aggregates. So the isothermal measurements for TAM can detect these denaturation and aggregation kinetics at temperatures below the TM. So before it starts this melting process or this unfolding process, we can at least get a snapshot of what the kinetics are and how it's happening. You can do it in 3 to 5 days. And if you start looking at the typical experiments that happen when you look at shelf life studies, you're talking weeks and you're talking months sometimes. And the assays that are used, even in the HPLC systems, sometimes are not that straightforward. It provides a sensitivity to detect heat signals in very small volumes, less than 1 milliliter. But then you can get it up in high concentrations and have samples that are at least greater than 100 milligrams per mil. This is a departure from what happens with standard analysis when it comes to shelf life studies. And it provides sample throughput. The TAM, with its up to 48 samples, you can assess multiple buffers and excipient conditions simultaneously and get an idea of a matrix situation that lets you make a decision rapidly and accurately. Enthalpy screening by ITC is a very powerful technique that rapidly and accurately characterizes the thermodynamic signature for any kind of binding reaction of drugs or biopharmaceuticals. The idea that isothermal microcalorimetry with a TAM4 has the possibility of revolutionizing the characterization of biological formulations. This is one of those areas that this kind of data is going to emerge and make a difference in the way we analyze the stability of biopharmaceuticals, especially high concentration biopharmaceuticals. The Affinity ITC Auto, the instrumentation for the enthalpy screening, it's innovative, it's a reliable platform that allows the user to set up enthalpy screen experiments very easily and rapidly. And the idea that software is there so that you can analyze multiple files in a batch process and compare it quickly and easily and make the decision that you have to make on each individual candidate. The TAM4 is an industry standard for isothermal microcalorimetry. And with its sensitivity and flexibility, you can do many, many things on it. But the idea that you can look at the rate of denaturation and of aggregation in a very short period of time when compared to the standard shelf life studies that are typically done, then isothermal microcalorimetry becomes a tool of choice for characterizing the kinetics that may exist in these high concentration biopharmaceuticals. The publications that you can look at that I based most of the data that you see in this presentation come from what you see on this page. Arnie Schoen and Ernesto Freire have been the pioneers in describing the enthalpy screening, how to set it up, what kind of instrumentation to use, and how to analyze the data, and the power at which it correlates with those standard titration data that has been published in the past. For the biopharmaceutical stability, there are several publications over the last few years that have touted the capabilities of looking at isothermal calorimetry. The idea that you can model the data and look at the kinetics of denaturation and of aggregation is something that isn't particularly new, but using this kind of data to then predict high concentration biopharmaceutical stability is something that may revolutionize the way we characterize those particular molecules. So I hope you've gotten something out of this particular webinar. It's something, if you need additional information, please feel free to call TA Instrument information line or any of the application scientists. Thank you for your time today.
Thank you, Dal, for that informative presentation. A recorded version of this webinar will be archived and available online through TA Instrument's website. We will now begin the question and answer segment of this webinar. Please submit your questions through the Q&A window. We will do our best to answer as many questions as possible. Okay, Dial, I'd like to go through some questions now that have come in from our audience. The first one is, what are the best troubleshooting techniques when experiments are not reproducible?
Well, Ed, when I saw this one come in, I thought in two different ways. First is troubleshooting the hardware. And when you have complex instrumentation, you want to be able to have something where you can troubleshoot it and make sure it's working correctly. So one of the things that we try to do with, especially with isothermal titration calorimetry, is a technique called water-water titration. And this is something that looks at the sensitivity of the instrument, how clean the instrument is, all of the things that are necessary to determine whether or not your hardware is working correctly. So if there's any question about the hardware and how it's working with a ITC experiment, that's one thing that can be performed that will give you a good idea that your hardware is working correctly. The second part is experimental design. And of course, experimental design has a lot of parameters in it. One of them being your reagents. How stable are they? What's the concentration? You know, is it a specific pH? Is it a specific formulation that's having an influence on whether you're getting irreproducibility or not? And so the idea would be then to use all of the data at hand that you have on your particular reagents to determine whether or not you are on an edge of stability for those particular proteins, and that's giving you this inconsistent result in an ITC. So both of those parameters, or both of those approaches to inconsistent results, are things that I would do and try to leverage as much information as I had about hardware and about reagents for troubleshooting.
Okay, great. Thanks, Dial. Next question. Is the TAM4 okay for using gel samples?
And the answer to that is yes. I wasn't able to spend a lot of time talking about the flexibility of the TAM4, but one of the powerful things that TAM4 brings to research and a characterization lab is its flexibility. We have different ampules, glass, stainless steel, Hastelloy. All of those allow you to think about the different samples that you can put in them, along with the different volumes down to 1 milliliter, 20 milliliters, 125 milliliters. All of those say that you can put those kind of samples in there or in the ampules and be able to get good data. So the idea that gels can be analyzed, I think, looking at stability of gel structures, as well as looking at reactivity on gel surfaces, are things that can be looked at in a TAM4.
Okay, great. A couple other questions that have come in, Dial. Next one. What is the total amount of protein that is actually used per ligand in the enthalpy screen experimental design?
Well, you know, this particular question comes up, and it's always there are some contingencies around it. But the idea is that you are going to have your macromolecule or your target in your injection syringe at a fairly high concentration. But when all things are said and done, if you're looking at things like antibodies that bind or drugs that bind, is that typically for enzyme targets or molecular protein targets, is that somewhere about 5 micrograms total can be used, and you have a complete enthalpy estimation from an enthalpy screen. Now that'll vary a little bit. It'll vary on how much heat is generated when you have a single titration of, say, 2 microliters, depending on your concentration, depending on your drug concentration as well, your ligand. But the idea is that you're not using what typically in a full isothermal titration experiment is that you can use up to a 0.5 milligram. And so you're talking 500 micrograms, but here we're talking about using 5. So you're getting at least an order of magnitude or 2 down from what is typically used for a full titration. And that has its attractive nature. Some people, some research labs just don't have the reagents where they could do a full titration. So now they can consider an enthalpy screen.
Okay, great, Dial. How about one more question? Comes to us, what other techniques have been used in conjunction with the enthalpy values to approximate the thermodynamic signature?
Yeah, that's a question that came up in several discussions that I've had recently about enthalpy screening. Is what do you use to get the other parameters like delta G, the binding constant, those things? Well, typically they're, like surface plasmon resonance, SPR. It's a very fast technique, uses very little protein, and gives you a good estimation of the KD. And so by doing that and coupling that with the delta H measurement for the enthalpy screen, you can then solve that thermodynamic equation. Another one that's used is enzyme inhibition assays, where you get an IC50, and so that or kinetics of inhibition. And if you use that data to then understand what the binding kinetics are, you can understand the energetics of the total binding, and then relate that back with the enthalpy value you've determined by an enthalpy screen, you then get the thermodynamic signature. So there's no restriction as to what it is. You want something that's going to give you a value that you can then solve that thermodynamic equation and be able to assign reasonable and approximate values to the delta S values that you might get, the delta G values that you would get, so that you understand what that thermodynamic signature is.
Okay, great. All right. Well, thank you, Dial, for answering those questions. And I would like to just mention to everybody that any other questions that we did not get to answer here, we will answer by email and follow up in that way. I also would like to remind everybody that the webinar will be available in 24 to 48 hours as a recording on our website. So if you would like to share it with colleagues or watch it again, please go to tainstruments.com and you'll be able to view the webinar again. Thank you, everybody, for joining us.
Yeah. Thank you, Ed.